Hi everyone, this is Dr. A. So we're going to do a quick overview of molecular diagnostics. So by no means this is thorough at all. It's just really skimming over it so that you get some of the gist of it and some of the basic information related to molecular. Okay, so let's start with a quick review of nucleic acid structure. So DNA obviously contains genetic information, and that genetic information is coded by nucleic acids. Nucleic acids are called polynucleotides because they're made of a sugar molecule with a phosphate group and then a nitrogenous base. And they're then arranged in a linear fashion, so lined up, right? And they produce a polynucleotide. And so you have two types of the nucleic acids. You have purines and pyrimidines. So purines are your adenine and guanine, so your A's and your G's. And your pyrimidines are your thymine and cytosine and your uracil, so your T, C, and your U bases in the code. And then DNA and RNA will differ in the sugar. For one, DNA is a deoxyribose sugar and RNA is a ribose sugar. So one of the ways to think about the genetic code with the A, C, T's, and G's is to think of how the internet code is, as in how information is transmitted. It's actually transmitted in a binary code with ones and zeros. And just with ones and zeros, you have this infinite combination, it seems like, of, that can code for almost everything that is on the internet. So our stuff is coded instead of binary with the 4 bases, and so the combinations are even bigger than with binary code. All right, let's talk about the structure of DNA. So the primary structure of DNA is considered of a sugar phosphate backbone that is made of the covalent bonding of the phosphate groups and the 3rd and 5 carbons of the nitrogenous bases. So that's and there's 2 strands. There's 2 backbones, 2 strands. And the secondary structure of DNA is that the strands are complementary. That means they're matched. So that wherever there's an A, it would match to a T. Whenever there's a C, it would match to a G, and they're matched up like that, and vice versa. Whenever there's a T, there's an A on the other strand, and whenever there's a G, there's a C on the other strand. Also, those strands are anti-parallel, so instead of being like this, they actually are like that. They go in opposite direction. The nucleotide bases face the inside of the strands that are parallel, so they're on the inside part, and the backbones are on the outside of the molecules. And they always pair one purine with one pyrimidine, but again, it's very specific pairing. It's always A with T and C with G. And then so the final structure is a double helix, twisted 2 strands of the DNA molecules. So this is what it looks like. So in purple, the sugar phosphate backbone is represented with the 2 strands twisting like that. They're in an parallel arrangement. So one's going in one direction, the other one's going in the other direction here. And so you can see the phosphate groups are illustrated here in green, the sugar in purple, and you can see the direction by looking at where the oxygen molecule is. You can see one's going one way, the other one's going the other way, and then you have on the inside of this double-stranded molecule, you have your bases shown as to how they pair up. All right, and so these pair with hydrogen bonds right there, so they do stick together, but they are easily separated so that the code can be read, as we're going to talk about later here in just a few slides. So the structure of RNA is single-stranded to begin with. Other than that, it is very similar to DNA. Again, the sugar is a ribose instead of a deoxyribose. There are 3 distinct forms of RNA. There's ribosomal RNA, which is 80% of the RNA that's made. There's messenger RNA, which is the RNA that codes for protein. And then there's transfer RNA, and the transfer RNA brings the amino acid, it transports amino acid molecules. The messenger RNA nucleotides are recycled and reused. And the bases in RNA are adenine, guanine, cytosine, and uracil. So uracil is similar to thymine, and it replaces it in RNA. So chromosomes, all DNA is contained in chromosomes. And chromosomes, as you've seen them in the little X formats and stuff like that, are actually supercoiled DNA strand, really supercoiled chromatin, which is DNA strands. They're coiled around histones, which are little proteins around which the DNA. So it's really tightly packaged DNA is what chromosomes are. They in a cell they don't always exist in the format of chromosomes, but when the cell copies itself and starts to divide, then you can see it in those chromosome format. The centromere joins the two arms of the chromosomes. There's the P arm and the Q arm of the chromosomes, and so it's joined in the middle by the centromeres. There are 23 pairs of chromosomes. 22 of them are the autosomal ones, and the 23rd pair is the sex chromosome. And the way it goes is, for example, for chromosome 1, for the first pair, you have one chromosome that's from mom and the other chromosome that's from dad, and that's the pair of chromosome 1. And then chromosome 2, you get one from mom, one from dad, that's the pair of chromosome 2, et cetera, and then your 23rd is your XY or XX chromosome. Genes, the human genome has 23,000 genes, and they each have a specific location on the chromosomes. So some genes may be located on chromosome 1, while another one may be located on chromosome 2. Another gene might be sex-linked chromosome, etc. And genes, if you will, are a section of the base codes, and it usually it's a set of instruction or codes for a specific instruction of something to be made, turned on, or turned off in the body, or it's a specific code for protein. Now, only 2% of the genetic code is actually codes for protein. The rest of it has to do with control of what's made when, etc. So again, here's a representation of a human karyotype. So you can see again the 22 autosomal pairs, and then with the 23rd, the sex pair, you can see the two arms of the chromosome each time right there, joined by the centromeres. And then you have, again, you'll get one of these from mom, one of those from dad, for each of these pairs. So this is a matching activity if you're in a neopod in the class, and you can do that one, so match the word with its definition. And then we are going to talk about DNA replication. So replication is making an exact copy of the DNA. This happens when cells need to be duplicated, and so from one parent cell you will get two daughter cells that have the exact same genetic material as the parent cell had. So this is where, for example, you're replacing skin cells all the time, and so your body's constantly making more skin cells, and from the parent skin cell it's making more daughter skin cells. So that's just an example. DNA replication is a complicated process. So the molecule helicase has to unwind the DNA. The two strands has to separate because they're twisted, so it has to open it up and unwind it. And then as it unwinds, and one of the strands will serve as a template. Well, each strand serves as a template actually. And RNA primers are needed to start the process. And one of the strands is going to be copied continuously because it's read in the same direction, so it's just add, it just keeps going. Where the opposite strand has to be read, if you will, backwards from the way the strand is opening it up. So it is copied backwards in sequences, and then the sequences have to be linked together, and ligases will attach these fragments of DNA. At the end, you have two identical copies of the DNA, but in each copy strand, you actually have half the parent strand, and then you have the other half is to copy the new half of the strand. The factors that are needed for DNA replication are energy, and the final product again is an exact copy of the parent strain. But again, if you start the two strains that start at the beginning, and one of them ends up in one copy, the other one ends up in the other copy. Then we have the process of transcription. That is the production of RNA from DNA in order to synthesize protein. DNA is the template that's read for the transcription into RNA. It uses RNA polymerase to generate that RNA strand. Messenger RNA, which is a single-stranded RNA molecule, is a template for the production of proteins. It is then fed into the next step into ribosomes to make proteins. DNA can also be transcribed into ribosomal RNA. Ribosomal RNA will be incorporated into ribosomes and is involved in the formation of peptide bonds. It's linking those amino acids by peptide bonds to make the proteins, because ribosomes are where proteins are made. And then your transfer RNA, they pick up and transport amino acids, and each transfer RNA molecule only transports one specific type of amino acid. And then translation is the process of protein synthesis, so it's the process of translating the messenger RNA into an actual protein. So the messenger RNA is fed to the ribosomes. Ribosomes produce protein. They're located in the cell cytoplasm and in the endoplasmic reticulum. The transfer RNA then brings the amino acids to the ribosomes and then add them to the growing chain. The way it knows how to add is it reads the nucleotide sequences in groups of 3 on the messenger RNA. And so this disk here represents, for example, how it's read. So if the first letter is a G, then it's a C, and then it's an A, or a C, or a U, or a G. So this is 1, 2, 3. So if it reads D's, so it reads, let's say, G, C, C, then it would match the transfer RNA that is carrying alanine, and alanine would be added to the strand. And then, but if it read G, G, A, it would add glycine next. And it just reads that code on the messenger RNA, a codon on a time. So a codon is 3 nucleotides, so every 3 nucleotides is read as a codon, and an amino acid is added. Of course, we could also take a little moment here to say that what happens is then if there is a mutation and something is changed, then this could go from coding for alanine. For example, if the C was changed and became a G, it would go for coding for alanine to coding for glycine, and that's how you can have mutations. All right, so again, another little matching exercise if you're in the class in the neuropod activity. So next we're going to talk about restriction enzymes. So this was a great discovery that basically allowed us to start genetic engineering and stuff like that. So restrictions enzymes are naturally produced by bacteria, and they're made to cut up DNA, and they can cut DNA at specific base sequences. There are really a defense mechanism of bacteria against foreign DNA, but we are using it, and what we found is each restriction enzyme recognizes a specific sequence of bases. It's usually a palindrome, meaning it can be read forward or backwards, and it reads the same way. And it recognizes it and cuts right there. And when it cuts, it breaks the hydrogen bonds between the bases and the phosphoester bond of the backbone. So it completely cuts the molecule. One of the cool things that allows for genetic engineering is if the DNA is cut by the same restriction enzyme, then even if they're from like different species, those pieces can be linked together because it's the same palindrome, so it's the same sequence it recognizes, so those will be complementary and stick to each other, and it's pretty cool. This allows for DNA identification. We're going to talk about that in a minute. But basically, each of us, if you incubate the DNA with restriction enzymes, it will cut it in a different way. Okay, so there's a video here. I'm going to link it at the bottom a little bit more explaining how restriction enzymes work. It's very interesting. And if you're in my class, answer this question. So just out of curiosity, can you name me a molecular diagnostic test or a technique that you've heard of? Next, we're going to talk about RFLP, so this restriction fragment length polymorphism. And this technique is used in the definitive identification of individuals, so a lot of like crime scene investigation and that kind of stuff. How it's done is first you use restriction enzymes, so an endonuclease, and you incubate that with your sample, and it will produce DNA fragments of different lengths. And again, the way the restriction enzyme cuts up your DNA is unique for each individual. So the fragments, length of the fragments generated are going to vary by people. And then those fragments are separated through gel electrophoresis. And then the southern blot process is applied. I'm going to talk about that in a little bit, and I'm going to show you a diagram of how it's done. But that allows you to basically visualize the separated bands of DNA via X-ray, and you get a pattern. And then, so for example, in this one, if this was your crime scene sample and then these were all your suspects, it is matching this one right here, and so you would identify then and match it up and identify that person as the criminal. So every person has a unique separation pattern using RFLP here. So again, here's the process. You get a blood sample, you extract DNA from the blood cells. It's actually usually amplified also. And then you use the restriction enzymes to cut up it will cut up DNA. And then you use electrophoresis to separate the DNA fragments into a gel. And then you use the fragments are blotted onto paper. So that's the blotting technique. So it's transferred from the gel to a paper. Okay. And then the radioactive DNA probes will bind to the specific DNA fragment that's on that paper that transferred, and then wash off the excess probes, and then an X-ray film is taken of the radioactive probes, and it generates a specific pattern of sequence that you can then, it uniquely identifies individuals, and then you can match a suspect with an unknown and just see if there's a match. PCR, so this is how DNA is amplified. So it's polymerase chain reaction. So its sole job is to take the little bit of DNA that you collected, that was collected from something, from a crime scene, from blood, from cigarette butt or something like that, and amplify it so there's enough of it to separate through electrophoresis to see those bands and stuff like that. So it uses thermal cycling, so it's a cycling of temperatures, and in the end there are many DNAs of copies that can be produced. So from one strand in as little as 30 cycles, you can produce, 1 million strands of the same copies of that DNA. The steps of PCR include first a denaturation step. So you heat the sample to 94 degrees to 98 degrees Celsius, so it's just shy of boiling, right? So it's really high, and this is we're in Celsius, right? Not Fahrenheit, okay? So as the DNA strands are denatured, they will separate out like that. And then the annealing step is you have to cool it to 50 to 65 degrees Celsius and incubate it with primers, and those primers will anneal or bind onto your separated DNA strand. Those are usually RNA primers. And then the extension is polymerase, so usually DNA polymerase. It starts adding the bases, the ACTs and Gs, as it copies the strand, so like in the process of replication inside your cells. And so you had your two strands that were denatured, so separated, the primers annealed on the beginning, and then you have extension, which happens at 72 Celsius, heat it up just a little bit more, and add the ACTs and Gs, or add it to the strain. And then it starts over again, basically. So that would be one cycle, would be denature, anneal, extension. And so you have a copies made, all the little bases are added, and then you denature again. So now you have the new one with its copy, and then they separate. You anneal and you add bases, and so basically you do that so many cycles, and then usually there's a final hold after the adequate number of cycles that preserve the DNA produced, and that one is at colder temperatures, 4 to 15 degrees Celsius. So basically, each cycle doubles the amount of DNA. And so I'm going to show you the video, but basically you go from 1 to 2, 2 to 4, 4 to 8, etc., etc. So this video is a good visualization. I'm going to play it. Granted, I don't expect you to be able to hear
Polymerase
it
chain
very
reaction,
well,
or
but,
PCR,
it's showing,
uses
so
repeated
DNA,
cycles of heating and cooling to make many copies of a specific region of DNA.
So you can see the little temperature gauge here.
First, the
it's
temperature
it
is
up,
raised to near boiling, causing the double-stranded DNA to separate or denature into single strands.
Of the strand. And then we're going to drop
when the temperature is decreased,
so
short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence.
The temperature's going to come up, and you're going to add
the primers bracket the target sequence to be copied.
Part of the, the
at a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle.
That's one cycle. Then you heat it up, you anneal again. So heat it up, you denature.
In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies.
Okay, so every time you cycle like that, you can see it doubles. So you go from 2 to 4.
After 3 cycles, the target sequence defined by the primers begins to accumulate.
And so the target sequences get
after 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Until you can get literally like 1 million copies. Sorry, didn't need to restart that. Okay, so again, visually represented, here you go with the doubling going on. So from the beginning, first it doubles, so you get 2, then you get 4, then you get 8, then you get 16, and so on, and 32, and 64, etc., etc. Okay, so all it does is amplify DNA. Then what do you do with it? It's up to you after that with the different techniques that exist. So one of the other tools that we have are DNA probes. So DNA probes are small segments of DNA. They are labeled usually in some way that can be detected. A lot of times it's fluorescent molecules. And then they're attached, a probe is then incubated and can attach to the sample DNA. So in this little diagram here, you have your sample DNA is here, and it is like denatured here where it's separated, and then the probes are allowed to hybridize. So why is it hybridized? Is because then what you have if it's complementary, it can bind to it and stick to it. And so that's hybridized because the end product is half the sample and half the probe. And so it's a hybrid. Okay? And the probe part is labeled and can be detected. So DNA probes are very sensitive and specific. They will only stick or anneal there to complementary sequences. So if the sequence doesn't match, the probe will not stick, and then you have a wash step usually, and it would wash any inbound probes, and then it just, there would be no reaction and stuff. They're often used to identify specific genes or chromosomes and stuff, or specific genetic sequences. And so, yeah, they can be used for tumors and all that. So some of the applications then are, the first one is DNA microchip. We're going to talk about this detects gene mutations. It's mainly used in research. The way it happens is DNA is put onto a chip surface that can be, usually it's a glass plate in plastic. And the DNA that is loaded on there contains thousands of single-stranded DNA pieces. And they make up the entire normal gene of interest. So we're just looking at one specific gene at a time. And all the pieces of the normal gene of interest are all tagged with the same color, a fluorescent color, let's say in our example, green. So all the normal stuff is green and it's fixed, and they're single-stranded, so it's one half of the DNA is fixed on that chip surface. Then the sample DNA is denatured into single-stranded DNA, so we get our single strand and cut into pieces and tagged with a fluorescent dye that is a different color than your normal gene. And so for here it could be red. So all the pieces are tagged with red, and then you take your single-stranded DNA from your sample in the single-stranded DNA that's on the chip surface, the normal one, and you incubate them and allow them to hybridize with the normal single strands there. And so anywhere where it's complementary, it will stick. And if they, so as long as the sample genetic sequence is normal, they will bind, it will be complementary and bind to the normal strains. But it will not bind anywhere where there's a mutation that's present because the sequence has changed and it's no longer complementary. And so that would generate a color difference that you can see in that gene. So you could see a pattern of, some green red mix, but then maybe an area where it's just one color. And so that is one way DNA microchips where you can detect the presence of genetic mutations. Another application is the DNA microarray. So it uses a single microscope top slide with a lot of wells and stuff in it. And it uses colored fluorescent nucleotides. So again, little probes, and they are labeled with different fluorescent dyes. And this is used to assess the tumor expression of certain genes. So as these things are incubated, the normal genes with the tumor genes, single-stranded again, and they combine and stuff. They hybridize, the probes hybridize to the sample on the microarray, and any gene that are more active will generate a brighter fluorescent color. Anything that's less active, a dimmer, and then depending on the type of genes, which one's tagged, it will be one color or a next. And so this is kind of what a microarray would look like. Obviously, this is a pattern is really hard to interpret with a naked eye, and so maybe some of them is like no expression. There's black. Some of them are brighter. Some of them are dimmer. Some of them are yellow, orange, red, etc., green. And so usually computers interpret these patterns for you, but you are looking at usually tumor gene expressions, increase, decrease, absent, something like that, normal, whatever. An application, a microarray application, is the BioFire. Very interesting. Again, I'm just going to link this one, but it's a combination of PCR and probes and microarrays, and the BioFire uses these to run panels on respiratory, the two big ones are respiratory and GI, where you can take sputum, like respiratory secretions, and run this on it, this BioFire panel, respiratory panel on it, and then you can identify like all the different possible respiratory infections. Like you can test for all of it at once, whether it's viral or bacterial, whatever, because it just looks for the genetic sequence of what's in the sample. And so anyway, very, very neat technology, wide application. And again, I will link it below. It's definitely worth watching. The next technique is Sanger sequencing. So it is used to determine the sequence of bases in DNA. So what's the order of those A, C, T's, and G's? First, DNA is fragmented, and then it uses different colored fluorescent labels on each nucleotide base of each, it's usually on the end base of each fragment. And then it uses capillary electrophoresis to separate these fragments by size, with the smallest one coming through first and then larger and larger and larger. And it identifies the labeled nucleotide at the end of each fragment and is able to read the sequence in that way. So basically the way it's fed through is, so again, they're broken up into pieces. These are all the pieces, and the way you put the pieces in sequence is just through capillary electrophoresis. But before you put them in sequence, you tag them, and again, only the nucleotide at the end will be actually the one that has a fluorescent dye, and each nucleotide has its own color. So the A's in this example are orange, the T's are yellows, the G's are pink, and the C's are blue. And then you separate them through electrophoresis, through capillary electrophoresis, where the smallest ones will come through first, and then it goes bigger and bigger and bigger and bigger. And then you literally just read the sequence of the colors that are coming through, and it's reading it as A, T, G, etc., and it reads the sequence and prints the sequence out for you. Again, so this is another one that shows Sanger sequencing. It's a really neat illustration. So again, I'm going to link it below for you guys. And then, so Southern Blot, again, just explained it, but let's go over it again. It detects specific DNA sequences. So Southern Blot is used for DNA. The restriction endonucleases, so those restriction enzymes, are incubated with a sample, and they form various strands size of DNA fragments. This is unique to each individual, how your DNA gets cut up by these restriction endonucleases. Then you do electrophoresis to separate the fragments by size. Then the DNA is transferred to a filter membrane and then baked actually to make it stick. And then DNA probes are added to the filter membrane, and those are used to visualize and identify the fragments. The pattern is examined. The pattern is unique to each individual, and then it can be matched to, again, usually a pattern of the sample that you're trying to identify. So, again, electrophoresis, been separated, nitrocellulose filter is used, blot it, then the DNA has been transferred to the filter from the gel, then you take the filter, hybridize it with nucleic acid probes, and then the way the probes are labeled, and so then you can visualize the separate, usually like if it's radioactive, then it's using X-ray and stuff, and does like the patterns and stuff. Sorry. Okay, so basically, what do we use this to? What are the diagnostic applications of molecular? So to screen and detect genetic diseases, and you can look for an abnormal number of chromosomes, so triploidity, so people like Down syndromes and all that, where there's an extra chromosome at one of the chromosome pairs. Also look for abnormality in structures or mutations in the chromosome's genetic code. It can be things like deletions in the sequences, duplications of chromosomes, translocations, which is where a gene on one chromosome like jumps to a different gene, so different chromosomes and stuff like that. There can be inversions and stuff like that. Rings can be made with the chromosomes, so there's all kinds of abnormal structures that can be identified that way. Of course, as we talked about too, you can, with the genetic mutations, you can look at the mutations within tumors, and those can often be used to guide different therapies. They know that with certain genetic mutations in certain cancer cells or certain types of cancer, that they respond better to treatment X or Y or whatever. And so that can help guide therapy for these patients. And then, of course, infectious diseases, identifying bacteria and viruses, but also genotyping of these different bacteria and viruses so that you can track them. So this is where epidemiology comes in, just like we've been tracking the coronavirus, and it has already mutated and changed. They know that because they're genotyping and they're seeing how the sequence is changing over time. You can also use molecular diagnostics to do viral loads. Viral loads, the biggest application for viral loads is going to be in HIV testing for HIV positive individuals to check to see what their viral load is, how much of the HIV virus they have. So anyway, that's all the diagnostic applications. Then if you're in class, you're going to have a few poll questions to be thinking about. So tell me what its restrictions enzymes are used to do. Then you're going to tell me in the applications which technique is used to amplify DNA. If you're simply on video, you can try to answer that mentally in your head. Next, you're going to tell me which technique is used to look for mutated DNA. Go. And next, which technique is used to identify individuals. And there you go. So if you're in class, you are free to ask a question. If you're not, you can always drop a question below. Again, this is a very quick overview of all the just basic things, basic tools of molecular diagnostics as a part of clinical chemistry. So thank you.